and now we're going to have more discussions on from Dr. Kang and Dr. Kavanaugh about phenylbutyrate and similar drugs.
Good afternoon, everyone. Can everyone hear me well? It's good? Okay. Yeah, so today I'm going to talk about our findings by comparing for phenylbutyrate and other small molecules in SLA mutations. Let me go my... How can I advance? Yeah. Yeah, so actually I reflect a little bit before I present this work. Today is my 7th talk for this symposium. So I'm very thankful to Amber for the invitation. I also feel a bit embarrassed, although I have progressed, but I'm so happy for the gene therapy, all those good things. So one thing I can say, we brought Ravicti to patient. So this is something we're very happy. And thanks to hero mom, like Amber and Terry Jo, the dear friend and Zach and Scott also, and now we move on to GABA A. So at the same time, we have published also co-authored 12 manuscripts, and we have 1 now in press, 3 under review revision. So we have done so much, but what does it mean to parents, to patients? Have we done something impactful? So that's what I always ask myself. So today, I'm going to give you a brief update on 3 things. So first, I would like to talk about PBA and other small molecules in 32 variants. Second, I'm to have more in-depth study for PBA in cell and mouse model in some representative mutations. Number 3, we're going to talk about splice site mutation because we have a missense mutation, nonsense mutation, deletion, like copy number variation, but no one have ever reported splice site mutation. So this is very important, especially given the gene therapy Ravicti showing like a promise. So first, I'm going to talk about our focus on 5 drugs. Previously, we have tested about 20 molecules. That's why we got Ravicti. Now we have compared Ravicti and Butyrate, TUDCA, which is chaperone, Amber just mentioned, or Celebrilla is a ER stress reducer, and SAHA is a HDAC inhibitor. So as this one, I saw a few speakers showed, we published 5 years ago. That time we look at 22 mutations. We identified either complete or partial loss of function for GABA transporter. Since 8 years ago, we have already started AI prediction. That's before that, so we have done a lot. This is just the show example. The first mutation we have published in 2019. We use AI, like machine learning tools. So in our lab we use AI tools, and after that we move on to cell model and then mouse model. And so this is most recent work. We use AI tools to look at the stability of this mutant protein across all these variants. If not all, majority of these variants show reduced stability. So this is Lindsay's children, and then we compare with the previously reported H375V. You see both show reduced stability. And in our lab, as you know, we study use cell and mouse models. In cell, we have HEC cells, we have patient-derived neuron and astrocytes. Previously, we have identified PBA Ravicti increase GABA uptake in HEC cells, and then in neurons, and then in astrocytes. So that's why 2020 we have a first talk with Dr. Zach Greenspan to have this trial. Right now, because I have so many patients, and we continue. I have my R1s on this compound, so continue study. Today I'm going to show some data on 32 variants. So this is just a show across 32 variants. You see either complete or loss of function caused by these mutations. And first I show the homozygous condition. All these patients right now is heterozygous, so which means you have 1 copy is normal, and then you have 1 mutant copy. So how can we model that condition? So we use a heterozygous with mixed condition. We give a half gene dose of whatever and a half of mutant. within certain range, we still able to show like across the mutation you have a reduction. And then with the PBA for phenylbutyrate, which is Ravictin, we see cross mutation. You see, maybe have different magnitude. You see upregulation, about 20 to 30% of GABA uptake. We compare with the chaperone. That one has been used in ALS, which is combination of PBA and TDC at a 3:1 ratio. That study was published in New England Journal of Medicine, but later on we stopped. So we test the PBA versus TUDC. We actually see very comparable data of TUDC. So the chaperoning, so suggests probably chaperoning effect is useful for GABA A receptor, for GABA transporter mutation. And now we look at the butyrate. I know Michael has beautiful work on butyrate, and we also look at a lot at those same as PBA. We actually see smaller rescue for butyrate. And now we look at the SAHA. SAHA is like a HDAC inhibitor. We also see much either no effect or very small. And we focus on PBA and the chaperone TUDC because both show good effect. And we found out, so if we look at the total GABA transporter, you see both very similarly upregulate. And we also found with a different concentration you see in mutant patient condition, which is much more sensitive to the upregulation compared with the wild type. I think this is a very important figure here. So we differentially tag the patient allele versus the wild type allele. And now we look at the wild type allele because we want to see mechanistically how PBA or TUDC or butyrate modulate the GABA transporter, and so this is why you have increased uptake. So we found out which one is I want to see so you will see compare with the wild type, PBA only increase the upper band, but butyrate give you 2 bands. So the lower band is immature. So the immature band has no function. Actually will cause ER stress. So that means it's not how much protein you have, is the quantity of the protein. So basically you don't need a lot of protein, but you need a good amount at the cell membrane. You need those functional. So that's why if you give a cerebral, you have a very little increase in total protein. You actually have more GABA function. So it means the trafficking environment actually is quite important. You don't want to flax with a lot of protein. You actually want to have a good amount of protein. They can easily traffic to the membrane. So I think this is very important. And then we have a very in-depth study of PBA in 2 different mouse models. One represents complete loss of function, which is Maxwell's. The other one is A to the V, is a partial loss of function. So we actually did some immunohistochemistry. We found in mouse brain, you actually see very similar level of GABA transporter. Even A to the V has remaining function. So that means actually cross models, you will have a different phenotype. So the actually functional amount of transporter is quite similar between the complete loss function and the partial loss function. And then we have a very detailed study by isolating synapsome, which means you only plot like a neuron terminal from like a homogeneous brain tissue. And then we look at how much GABA transporter mediated activity there. And then we also pull out gliasome. So because we know GABA transporter is expressed both in neuron and astrocyte. So we look at both. And then compare both mouse models. You can see in both mouse models, you will have a reduced GABA activity at a neuronal terminal and the para-astrocyte regions. But if you block with SNMP5114 means GAT3, you will see similar phenotype. But if you block GABA transporter 1, you do not see much. That means the major GABA uptake in the brain actually is mediated by GABA transporter 1, by SLC6A1. It's the major one. And then we see PBA can actually rescue. So we treat animal for 7 days, 100 milligram kg for 7 days, and now we sacrifice the animal, and now we isolate the synapsome, and now we see the upregulation with PBA treatment. Compared with untreated, definitely the same. Same thing you see in synapsome and the gliasome. So both in the junction of neuron and astrocytes, you see increased GABA uptake with PBA treatment. And this is a work by Korea. He's going to have a more detailed talk on that. So this is an E-phase study. He identified, compared with the wild type animal, Maxwell's loss of function mouse model, you show prolonged decay. He will talk more, so I'm not going to waste time. And now with PBA treatment, not with 7 days treatment, but with 28 days chronic treatment orally, you see rescue of like normalized decay tau. So we think by look at the synapsome and the gliasome, we think PBA is more efficacious than butyrate in restoring GABA uptake in synapsome. This one, we have published 4 years ago. We show PBA can reduce seizures. This is not new. And here we compare PBA and butyrate. We both use 100 milligram kg for 7 days. This treatment compare with PBA. Butyrate is not as efficacious. Actually, in Maxwell's mouse model, we actually see increased seizure activity. In A to 8-way, you show a trend to reduce seizures, but it's not as good as PBA. I want to say our N number is not big, but the trend seems, I think we have confidence to say PBA is more favorable if we want to restore GABA uptake and reduce seizures, at least in that time window, like we did 2 to 4 months old, and with the dosage 100 milligram per kg. This is what we concluded. Now we have a paper is in revision, and also we look at behavior, because we just had a nice session. So many parents talk about the children's challenge behavior, and also we know seizures is not just one thing. How about the challenge your behaviors? For SLC, look at the 2 mouse models. We think locomotion deficit is the most robust phenotype. So this is why we look at mouse traveling and mouse hanging, mouse sleeping, those 3 things. The reason we choose these 3 metrics is because we have look at many neurobehavior tests. These only 3 things show very reproducible phenotype. So we rely on these 3. So this now the paper is already probably should be online available very soon. So we found with PBA treatment, either acute treatment 7 days or 28 days, with no dose, we see rescue. For travel distance, we see chronic treatment actually works better. For hanging, both chronic treatment and acute treatment can rescue. For sleep, we also see chronic treatment seems works better. So we have longitudinally monitored both mouse models from P0 to 10 to 12 months. We look at 1 month, 4 months, 7 months, 10 to 12 months. We found the robust become most prominent at 7 months old. But you see motion deficit at the very beginning, with direct reflex, you see at 5 days. But 1 month, we were already able to plot the difference, but at 7 months, it's much more robust. So we look at a lot of data, we look at 7 months. And third thing, I'm going to update our mini-gene project as Katrina's mutation. We think his mini-gene will actually take us, it's a massive endeavors. 2 level member full-time for several months, finally made it. So originally we look at a lot of different mutations, missense, nonsense, frame shift, same way. So this is the first splice site mutation. And so we engineered the construct by introducing intron 13, and engineered the mutation. So this is what we got. So with the CFP, just the marker ER, and we compare with the wild type and then with the splice site mutation, we clearly see the mutation have a lot of retained fluorescent signaling, which is very similar to actually Maxwell's and A2 edited way. This is the patient condition we mixed the wild type and mutant. You see kind of in between. And we have done some like GABA binding. The experiment is still ongoing, but the first that is promising, we see very similar to Maxwell's at the function level, but we still replicate and we don't know the response to Butyrate and PBA. We'll work on that. And Debbie actually is actually running experiment today. Yeah, so basically, I want to say, I have a good understanding because now when gene therapy is available, can everyone. No matter your missense or spread site or copy number variation, are you eligible? I think this is very important. And so we want to say, from the data we have so far, I want to say pharmacochaperone is a play pretty important role in mediating effect of PBA. Butyrate have a HDAC effect has been reported in other models to improve calculation or even some seizure activity, but I feel probably is much sensitive to the dosage and the age. But for SLC, I'm pretty confident PBA will work better. So I think this is important for parents. So take-home message from the update today is we think PBA is likely disease modifying. It's not just control seizures, as Amber mentioned. It's not a dirty drug. It modulates very upstream of the mechanism, and so everything happened, downstream is kind of like letter cells, because your body is very smart. They can adapt. When we fix the things at the very beginning, fix the protein level, a lot of downstream like circuitry level, behavior level could be very, I think, very, very modifying and change the patient outcome. And we think if your child have a splice mutation, likely your child will benefit from gene therapy and Ravicti like other mutations. And we think PBA for restoring GABA uptake and seizure control for these two will be more efficacious in our hand. I like this very much, Amber and Maxwell, because look at the fighting spirit. This is what we need, right? I like yeah. Yeah, so this people work with me and family. I'm very thankful for what we have made through, yeah. If you have any question?